Hello dear friends. So in this video we'll be discussing about the initiation of the process of translation. See, translation is the process by which the sequences or the codons in mRNA are translated into the amino acids in proteins. So first let's see how this process of translation of mRNA into the actual proteins is done. So for the process of translation to start, first we need an initiation complex. So let's see how that initiation complex is made. See, in the case of eukaryotes, we know we have ATS ribosomes. See, this ATS S is the Swedberg unit. This ATS ribosome actually has got two subunits. One is the 60S, that is the larger subunit. Other portion is the 40S or the smaller subunits. So when the body is done with when the cell is done with the process of synthesis of proteins, these two subunits actually separate. So in this cell, we don't have ATS ribosome as such. We only have 60S and the 40S subunits. Whenever we need to form the proteins, these two subunits should come together for the process of formation of proteins. So in the cell, now we have got this 60S subunit and 40S subunit. For the process of protein formation to start, first a specific type of transfer RNA, that is initiator tRNA or initiator transfer RNA, brings methionine amino acid to the ribosomes. Why this is known as initiator tRNA? Because in this transfer RNA, nitrogenous bases have got many modification in their structure. So this specific type of transfer RNA is known as initiator tRNA. So now we have to start forming the proteins. First, we have to make a ternary complex. This ternary complex is when this initiator tRNA binds with initiator factor 2 in the phosphorylated form. See, we have shown GTP here. So this structure is the GTP. This says our initiation factor 2. So ternary complex is when initiator tRNA that is already bound to the methionine here, binds to initiation factor 2 in phosphorylated form. So this whole thing is ternary complex. So how it's formed? It's formed when initiator tRNA that is bound to methionine combines with eukaryotic initiation factor 2 in the phosphorylated form. So how it's phosphorylated? This factor is phosphorylated with the help of eukaryotic initiation factor 2b. This factor 2b helps in converting GDP to GTP. So this is formed with the help of eukaryotic initiation factor 2b. So let's see what is the structure of IF2 or initiation factor 2. It has got three subunits actually, alpha, beta and gamma. These two, alpha and beta, these two when phosphorylated, they actually inactivates the initiation factor 2b. So when these are phosphorylated, this 2b is not able to phosphorylate here. So this is how the feedback structure is present here. So first let's make the ternary complex. For that, our initiator tRNA that is bound to the methionine amino acid combines with initiation factor 2 in the phosphorylated form. So this is ternary complex. Now we already had 40S subunit in this cell. This 40S subunit combines with anti-association factor, that is initiation factor 3, 1 and 1A. So for the synthesis of pre-initiation complex, these two, they come together. This thing 40s subunit that is already combined with factor 3, 1 and 1A. This combines with our ternary complex like this. This is the ternary complex. This is 40s subunit and this is our factor 3, 1 and 1A. And initiation factor 5 also comes here. So this whole thing 40s subunit combined with initiation factor 5, 3, 1, 1A and ternary complex is known as the 43S pre-initiation complex. These two subunits, that is 60S and 40S of the 80S ribosome, it has got three sites actually present in it. One is the P site, other is the A site and the other one is the E site or peptide site, acceptor site and exit site. So this ternary complex goes and binds with the P site present in the 40S subunit of the 80S ribosome. So what is the structure of 40S pre-initiation complex? This is the 40S subunit combined with ternary complex and with initiation factor 5, 3, 1 and 1A. So this is how the process of initiation actually commences. It commences with the synthesis of ternary complex and then 40S pre-initiation complex.